Hello everybody. So let us continue with what we were discussing. We are currently dealing with the chapter Human Reproduction, as you all are aware. And before we move into today's lecture, we shall again be going through the chapter overview or the roadmap as to what you are looking forward to. We are done with the male and the female reproductive system. We are done with gametogenesis, both spermatogenesis and oogenesis, and the reproductive cycle, the monthly reproductive cycle, that is the menstrual cycle in primate females, correct? So, and then in today's lecture, we are going to focus on fertilization, cleavage, and implantation. Following that, we will be discussing about pregnancy and embryonic development, and lastly, childbirth or parturition and nourishment of the newborn, that is lactation. Okay, so today's focus will be fertilization, the process of fertilization, followed by cleavage and implantation. A quick review of what we had learnt in the previous lecture. In the previous lecture, we had learnt about the process of egg formation. The formation of a mature female gamete is called oogenesis, isn't it? I hope you all recall that the process of oogenesis happens in the ovary, and it starts even before the birth in the fetal ovary. Then a part of it happens between birth to puberty, and then later at puberty there are meiotic divisions, isn't it? Then we had seen the stages of follicular development. We had seen how a primary follicle becomes a secondary follicle, which becomes a tertiary follicle, and which finally transforms into a mature Graafian follicle. And we had also discussed about the structure of the Graafian follicle, followed by when the Graafian follicle ruptures. We had discussed about ovulation, and then we had learned the different phases of menstrual cycle, which is the monthly reproductive cycle, which happens in humans and broadly speaking in all the primate mammals. Okay. So moving on to this lecture's, the focus of this lecture, which will be about a process referred to as fertilization. So how do you define fertilization? It can be defined as the process of the fusion between or the fusion of a sperm and the ovum. So the sperm is the male gamete and the ovum is the female gamete. So you all are aware that if this is the ovary, and very close to the ovary we had learnt the female reproductive system. We had seen that there was an infundibulum. This funnel like portion is the infundibulum, and then there are fimbriae at its free margin. So once the oocyte, the Graafian follicle ruptures inside, we had seen that the secondary oocyte is liberated, and then it enters into the fallopian tube, and then it makes its journey through the fallopian tube, and it reaches this broader part of the fallopian tube, which is called the ampulla, and it is here that it encounters the sperm. And this fusion that happens between the secondary oocyte and the sperm, which has come all the way up here into the ampullary region, is referred to as fertilization. So this is the ovary, and I had depicted the same in the last lecture also, the rupture of the Graafian follicle. So this is the rupturing Graafian follicle, and this is the secondary oocyte. So it is called as ovum in some places, but be very precise. The sperm merges or fuses with, not with the ovum, but with the secondary oocyte. Then what is ovum, you may ask? So as we progress further in this lecture, we will understand what exactly is the difference between the secondary oocyte and the ovum. Okay. So please all of you mark this to be a very important 1-mark question in your board exam. The definition of fertilization, I'm going to repeat it again. It is defined as the process of fusion of a sperm with an ovum. It's a very simplified definition given to you in your NCERT, so it shouldn't be a problem for you to remember it. Now, before fertilization happens, so where is this fertilization happening? I just told you that fertilization happens in the broader part of the fallopian tube, that is called the ampulla. So before that, what should happen? Before that, we are going to discuss about a set of pre-fertilization events. As the name itself indicates, pre-fertilization means something which happens before the process of fertilization. Now that involves arrival of the sperm and the transport or the movement of the sperm. So during sexual intercourse or copulation or coitus, we had discussed that there is insemination. If you remember, about 200 to 300 million sperm are inseminated or ejaculated into the female reproductive system, isn't it? Now, these motile sperm will swim rapidly and they will pass the narrow region of the uterus, which is the cervix. They enter the uterus and finally they reach their destination, which is the fallopian tube. And which part of fallopian tube? The ampulla. And who is in the ampulla? So, if this is the ampulla, and this is the infundibulum and the fimbriae, and this is the narrow part of the fallopian tube, which is the isthmus. So the egg or the oocyte has made its journey from the ovary, it has made its journey to the ampulla, and over here you can see the sperm are moving in, the sperm are being passed in, and finally one of the sperm successfully makes a physical contact with the secondary oocyte, which is sitting in the ampulla of the fallopian tube. So once again, this is a very important 1-mark question, all of you. So they may ask you, which is the site of fertilization in humans? You must be able to say that it is the ampullary region of the fallopian tube. If some of you have the older version of the textbook, it's mentioned as the ampullary isthmic junction, but that has been modified in the latest edition of your NCERT textbook. So please stick to this. The site of fertilization is the broader part of the fallopian tube and the longest part of the fallopian tube, which is referred to as the ampulla. Is that clear? So this is what should happen before fertilization. Simultaneously, the egg or the secondary oocyte and the sperm need to be transported and they need to reach the ampulla to ensure that fertilization is successful. Is that clear to all of you? Fine. Now, once the sperm has come closer to the oocyte, what exactly or how exactly does the oocyte look? So we saw that the Graafian follicle ruptures in the ovary, isn't it? So once again, I'm going to show you here. So if this is the ovary and if this is the Graafian follicle, and the Graafian follicle is rupturing here, and once the Graafian follicle ruptures, it's going to release the secondary oocyte. What exactly does the secondary oocyte look like? So now we are going to draw that diagram here. Now this diagram which I'm going to draw here, I want you to follow me and replicate it in your books, is a 3-mark question in your board exam. You may be asked to draw this diagram. The title of the diagram is Ovum Surrounded by a Few Sperm. Okay, so now the Graafian follicle, let me call this as the Graafian follicle has ruptured. And what has come out now? The secondary oocyte has been released. The process is called ovulation. Okay. Now let us see how exactly the secondary oocyte looks like if you magnify it. So if you magnify it, the secondary oocyte has a membrane. Now this thicker membrane that I'm showing you here, like any other cell, it will have a plasma membrane, and inside it has a nucleus. So in the nucleus you can expect the chromatin material, and between the nucleus and the plasma membrane you all know that there is a gelatinous matrix or the cytoplasm with cell organelles in it, correct? So I'm putting dots here to indicate the cytoplasm. So this is how the secondary oocyte looks like. Outside the plasma membrane there will be an additional membrane. So I'm showing you another membrane outside. I'm drawing this membrane in dotted line to create a distinction. I've shown the plasma membrane in solid line and I'm drawing this membrane in dotted line. And what is the name given to this membrane? This membrane is secreted by the oocyte itself. So the oocyte itself secretes this membrane. And this membrane is referred to as the zona pellucida. We had learned this in the previous lecture, I'm sure many of you remember that this additional membrane that is formed by the secondary oocyte is referred to as the zona pellucida. Now what should I label this inner membrane? This inner membrane, I'm going to label it as the plasma membrane. Like any cell, the secondary oocyte has a plasma membrane. Okay, then can you all see a gap here? This gap between the zona pellucida and the plasma membrane is referred to as, I'm going to write it here, it's called the perivitelline. Please draw it along with me. Perivitelline space. So three things we learned now. So this is the secondary oocyte, correct? Now this is the nucleus of the secondary oocyte, inside which you have the chromatin material. This is the cytoplasm, and this is the plasma membrane. The outer membrane is called zona pellucida. In between the two there is a space called the perivitelline space. Okay, now what lies outer to the zona pellucida? Remember that the secondary oocyte came by the rupturing of the Graafian follicle. So some of the granulosa cells or follicle cells are still stuck to it on its outer surface. So let me show them here. Some of these follicle cells are still attached, and they have certain microvilli like projections to them. See how I'm showing finger like projections here. So these are the follicle cells which are surrounding the egg, which are surrounding the secondary oocyte. So let me show the nucleus of these follicular cells, because they are also cells, they have a distinct nucleus and they are derived from the Graafian follicle, the membrana granulosa of the Graafian follicle. Now this cellular layer, this entire area which is made up of these follicle cells, this layer of cells is referred to as corona radiata. So please remember, this entire layer of these follicle cells which bear finger-like projections, this is referred to as the corona radiata. And now since we have to show the secondary oocyte is surrounded by the sperm, I will be showing a rough diagram of one or two sperm which have managed to approach the secondary oocyte. Okay, so you can show the sperm that is approaching the secondary oocyte in an attempt to fertilize it. So this diagram, you can refer to this refer to your NCERT for this diagram. Now since I had to explain, I have simplified the diagram for you. So what are the things which you must label in the diagram? I think you can label the sperm as well. Okay, so first we labeled the innermost covering of the secondary oocyte. We labeled it as the plasma membrane. Then we labeled this outer membrane called the zona pellucida. Then we labeled the space between the two as the perivitelline space. And then the outermost layer of cells, I told you that the secondary oocyte is covered by a layer of follicle cells. So this is called corona radiata. So corona radiata, for your information, in brackets I'm writing, I'm calling these cells as the granulosa or the follicle cells. Okay, and then you can see some of the sperm are approaching the secondary oocyte. So even though it is wrongly referred to as ovum surrounded by the few sperm, please remember it is actually a secondary oocyte. Is that clear to all of you? Okay, so this diagram is important from your PU board point of view. It's a 3-mark question, a diagram question, which you all must be thorough with. Clear? Okay. So the sperm has now approached the secondary oocyte. Where? Tell me, think and tell me. Yes, it is the ampullary region of the fallopian tube, isn't it? So one of the, like I told you, for fertilization to become successful, simultaneously the secondary oocyte and the sperm has to reach that spot, isn't it? Now once the sperm has come closer to the egg, And once the sperm makes its contact with the egg, it has to cross something called barriers in order to make its entry into the cytoplasm of the secondary oocyte. What are the barriers that it has to cross? It has to first cross, again I'm going to show you here a portion of the secondary oocyte. So this is the plasma membrane, and then I told you that outside the plasma membrane there is zona pellucida, and then I had drawn these follicle cells, the corona radiata layer with finger-like projections, isn't it? So let me put nuclei to the cells of corona radiata. And in between, so inside is the nucleus of the secondary oocyte, and this is the cytoplasm of the secondary oocyte. The sperm is coming from this side. So what all should the sperm cross before it enters into the cytoplasm? First, it has to cross the ground substance of corona radiata. Now, between the cells of corona radiata, there will be a ground substance, a matrix. So what I'm showing you here in stripes is the matrix. So first it should cross the matrix, isn't it? Then it should cross the corona radiata itself. Now suppose it crosses the matrix, and then it may bump into some of these cells. So it has to cross these cells also. So it has to overcome the ground substance of the corona radiata along with the cells of the corona radiata. Then you imagine where the head of the sperm will land up on. It will land up on this layer, which is referred to as the zona pellucida. So then it has to cross the zona pellucida. Yes or no? Okay. Then after it crosses the zona pellucida, where does it land up on? It lands up on the plasma membrane. This is the plasma membrane of whom? Plasma membrane of the egg or the secondary oocyte. So finally it has to cross the plasma membrane of the secondary oocyte. Is that clear? So you got a fair idea now. All of you see the diagram again. The sperm is moving in from here. First the sperm will land on the corona radiata. It has to overcome the ground substance of the corona radiata. Then the corona radiata itself, the cellular part of it. Then the head of the sperm has to overcome the barrier, which is the zona pellucida. And then finally it has to overcome the barrier, which is the plasma membrane. Only then it will successfully make its contact with the cytoplasm of the secondary oocyte. So this is the nucleus and this is the cytoplasm. So a total of 4 barriers must be crossed, and this is crossed with the help of a special structure in the head of the sperm, which is referred to as the acrosome. And this reaction, which helps the sperm to cross all these barriers with the help of the acrosome, is referred to as the acrosomal reaction. Do you remember in the sperm head we had shown the tadpole-like structure of the sperm? This is the head of the sperm and this is the midpiece of the sperm. In the sperm head we had shown a nucleus, an oval nucleus, and then sitting on top of the nucleus we had shown a cap-like structure, correct? Now this cap-like structure is what is referred to as the acrosome. Yes or no? So this acrosome is going to release certain enzymes. Please remember, it releases certain enzymes, and these enzymes are going to help cross these barriers. Isn't it? And it is said that the most important barrier which determines the fertilization, whether fertilization is successful or not, is the zona pellucida. So when the head of the sperm lands on the zona pellucida, this is what is going to determine whether or not the process of fertilization becomes successful, or the fertilization does not, or it ends up becoming or you can say that there is no fertilization, there is no formation of zygote at all. So who determines this, whether fertilization is successful or not? It is the zona pellucida. And where is zona pellucida present? So this is present outer to the plasma membrane. See, this is the plasma membrane, and outer to the plasma membrane you have the zona pellucida. Clear to all of you? So please remember, why do we call it as acrosomal reaction? We call it as acrosomal reaction because it is mediated by enzymes which are released by this cap-like structure, a vesicle that is sitting on top of the nucleus in the head of the sperm, and this is referred to as the acrosome. Clear? No doubts? Okay. And then finally, the sperm membrane and the egg membrane. Now, where does the sperm finally make its contact with? Once again, let me show you here. So, this is the plasma membrane. This is the zona pellucida. This is the corona radiata. Isn't it? So, now the sperm has to cross. First, it has to cross the corona radiata. Then it crosses the zona pellucida, and then it lands up on the plasma membrane of the secondary oocyte. So this is the nucleus of the secondary oocyte, and this is the cytoplasm. So now finally the sperm head has made its contact with what? With the plasma membrane of the secondary oocyte. So once again, here you can very clearly see this is the first barrier, the zona pellucida is the second barrier, and this is the plasma membrane happens to be the third barrier. So finally the sperm, and now what happens once the sperm makes its contact? So let me again show you the sperm here. This is the mid piece of the sperm, and this is the head of the sperm. The moment it makes contact with the plasma membrane, see how? So this is the plasma membrane of the secondary oocyte, and this is the plasma membrane of the sperm. So once it forms a contact, once the sperm head forms a contact with the plasma membrane, see how the plasma membrane and the sperm membrane, they're fusing at these points. They're becoming continuous. See? And once they become continuous, a bridge is formed through which the nucleus and the centriole. So this is the nucleus of the sperm. And behind the nucleus there is a centriole. What do you call this centriole? Proximal centriole. So both of them enter. What's inside the plasma membrane? Cytoplasm of the secondary oocyte. Yes or no? So this is what is mentioned over here. This is what is called sperm entry. So when does this happen? It happens here. Here it happens when the sperm makes its contact with what of the secondary oocyte? When the sperm makes its contact with the plasma membrane or the egg membrane. And then you can see there is fusion of the plasma membranes. PM means plasma membrane. Fusion between the plasma membrane of the secondary oocyte and the plasma membrane of the sperm. And then you can see how the cytoplasm of the sperm becomes continuous with the cytoplasm of the secondary oocyte, and the nucleus along with the centriole, the proximal centriole, will slide into the cytoplasm of the secondary oocyte. Isn't it? Yes? No doubts. Okay. So the sperm membrane, I'm reading out this sentence for you. The sperm membrane and the egg membrane fuse to establish a cytoplasmic continuity across which only the sperm nucleus and the proximal centriole, both of them, pass through. Nothing else does the sperm contribute. The sperm only contributes its nucleus and its proximal centriole. Did you understand what I'm saying? It does not, the entire sperm doesn't enter inside, neither does it contribute its mitochondria or any other cell organelle or cytoplasm. It only contributes its nucleus and the proximal centriole. So this is the sperm entry. This marks the process of fertilization. Finally, the sperm has crossed its three barriers: barrier 1, then barrier 2, and then barrier 3, and finally it has passed on its nucleus into the cytoplasm of the secondary oocyte. Clear? The entry of the sperm stimulates completion of meiosis II by the secondary oocyte, which now transforms into a haploid mature ovum. I've kept telling you that you should not call the secondary oocyte as ovum, and there is a difference between the secondary oocyte and the ovum. Okay, so now this was the secondary oocyte, this was the nucleus, and this was the cytoplasm. This was the zona pellucida, correct? And what was the outer layer? Corona radiata. Yes, and now the sperm has the membrane of the sperm and the membrane of the secondary oocyte have fused, and what has come in? The nucleus of the sperm has come in, along with what? The proximal centriole. See? So the zona, I have not shown zona pellucida here, but remember the sperm has crossed all these three barriers. Do you agree with me? So this is a simplified diagram here. This is the nucleus of the secondary oocyte. This is the nucleus of the sperm along with the proximal centriole, and this is the cytoplasm of the secondary oocyte. So what has entered now? The sperm nucleus along with the proximal centriole has successfully entered the cytoplasm. The moment this happens, the moment the sperm has entered, when I say sperm has entered, please remember the nucleus and the centriole. The moment that has entered, this nucleus of the secondary oocyte will complete what? Will complete its meiosis II. And now this cell becomes what? This cell becomes a, what does it become? Ovum. And it cuts off a tiny daughter cell. If you remember the schematic representation of oogenesis, when secondary oocyte completes its meiosis II, the meiosis II is unequal. You get a larger ovum, and a smaller cell that is cut off is referred to as the second polar body. Clear? So I will try and depict it for you over here, what exactly is happening. Okay, so again I'm going to draw the secondary oocyte, plasma membrane, zona pellucida. That's it. You imagine that there is corona radiata outside, and I'm going to only show a part of the sperm here, and what has already entered, the nucleus of the sperm, and what centriole? Proximal centriole. And what is this? This is the cytoplasm of the secondary oocyte. Now this is the nucleus of secondary oocyte. Meiosis II is yet to undergo, is yet to happen, so I'm calling it secondary oocyte. Now when the nucleus of the sperm has come in, this secondary, the nucleus of the secondary oocyte will undergo meiosis II, and it becomes a new nucleus now. Let me shade this also. So when it undergoes meiosis II, it forms a new nucleus here, and another small nucleus is pushed out with little bit of cytoplasm, and this cell that is formed, this tiny cell that is pushed out, and can you see that this tiny cell is occupying the gap? This gap, what did we call this gap? This gap, we called it perivitelline space. So the cell is so tiny that it is sitting in the perivitelline space. Now what is this tiny cell that is sitting here called? It is the second polar body. Okay, now this nucleus, which is formed newly now because it has undergone what? Meiosis 2. Now this cell which has a new nucleus now, you can call this cell as ovum. Are you understanding what I'm trying to say? Yes. So when did you call it a secondary oocyte? When it had this earlier nucleus with the zigzag line inside, you called it secondary oocyte. But after the sperm entered and after it underwent meiosis 2, it has a new nucleus now with it. And it has cut off a tiny cell in the perivitelline space. And now this huge cell with the new nucleus is called ovum. Did you understand what I'm saying to you? Okay, I hope the concept is clear. You can understand by again going through, by rerunning the video again. Okay, so at this point you should now remember that this huge massive cell that is there here is no longer called a secondary oocyte. It is now referred to only as what? Ovum. Clear to all of you? Okay. The nucleus of the sperm. What do we call the nucleus of the sperm? Now we call it as it becomes slightly enlarged in size. Because once it's inside the cytoplasm, now it has to gear up for the formation of zygote, isn't it? So now it is called the male pronucleus. And the new nucleus, what did we call that cell which now has a new nucleus? We called it as the mature ovum. And we call this new nucleus as the female pronucleus. They start approaching each other, they start moving closer to each other. Their nuclear envelopes dissolve. Once they come closer to each other, their nuclear membranes dissolve, and what happens to the chromosomes in them? The chromosomes will get mixed up. So the male pronucleus which came from the sperm has 23 chromosomes in it. And the nucleus of the ovum, which was formed by meiosis 2, now also has 23 chromosomes. So when the two nuclei come closer and when their membranes disappear and when the chromosomes mix up, it becomes 23 plus 23 is equal to 46 chromosome, meaning diploid set has now become 46. And now the cell which has 46 chromosome is no longer called ovum, it is called a zygote. So the cell is now called a zygote. It has how many chromosomes in it? It has 46 chromosomes. Did you understand the concept? So this is the last step of fertilization. It is called karyogamy or it's called amphimixis. And it is during this step that the chromosomes that are present in the male and the female pronuclei, they mix together and they form a zygote, because now the cell has how many chromosomes now? It has 23 plus 23, 46 chromosomes. Clear to all of you? Okay. Now, how exactly is the process of sex determination carried out in humans? Now, you know that in every cell of your body, you have two types of chromosomes. In that, I would like to specify or stress upon a specific chromosome called the sex chromosomes. These are also called allosomes. Humans have two types of allosomes, X and Y allosomes. Each cell of your body will have two of these allosomes. Okay, if you are a male, you will have in each cell of your body you will have X and Y. If you are a female, in each cell of your body you will have two copies of X chromosome. So that's what is mentioned here. The sex chromosomal pattern of human female is XX and male is XY. Males produce, now because males have two unlike sex chromosomes, XY. So if a cell which has XY chromosome were to undergo meiosis or spermatogenesis, then it produces two types of sperm. 50% of the sperm carrying X chromosome, 50% of the sperm carrying Y chromosome. So male is going to produce two types of sperm, and that's why the male is called heterogametic. Are you following? Now in female, if a cell which has two X chromosomes were to undergo meiosis, that means it's undergoing oogenesis in the ovary, then all the eggs will have only X chromosome. Why from 2X it's become X? Because remember, it's meiotic division. The cell participating in meiotic division is diploid with two sets of chromosomes, but the resulting gamete is always haploid. So the sperm also have, it's either X or Y, because the sperm are not diploid, the sperm are haploid. And why are the sperm and the egg haploid? Because the process of sperm formation and the process of egg formation involves not mitosis. What does it involve? It involves reductional division. The reductional division is nothing but meiosis. So because females produce all eggs with, or all eggs which are of the same type, because all the eggs have only one X chromosome, the females are said to be homogametic. Because males produce two different types of sperm, 50% of them with X chromosome, 50% of them with Y chromosome, we call males as hetero. Hetero means different, gametic means gametes, different types of sperm. Homo means similar, gametic means gamete, same type of gamete, similar type of eggs. So different types of sperm and similar types of eggs. Is this clear to all of you? What determines the sex of the child or the sex of the zygote? So you know that the sperm and the egg, both of them fuse together and you get a diploid cell which is called a zygote. Okay, depending on what type of sperm fuses with the egg, because it's the sperm that has either X or Y, isn't it? So it is the sperm which is the sex determining factor. So it's mentioned clearly here, the sex of the baby is determined by the sperm. To make it more clear to you, let me show it over here. So let me take a human male. I am not going to discuss about the other types of chromosomes. You might or might not have been introduced to this concept yet, but along with sex chromosomes, each and every cell of our body also has certain chromosomes called autosomes, which I am not bothered about now. I am only bothered about the chromosomes which are involved in sex determination, the allosomes or the sex chromosomes. So I'm only focusing on the sex chromosome. So a male has, unlike sex chromosomes XY, female has like sex chromosomes. So this is a cell, say in the seminiferous tubule of the male, and this is a cell, say in the ovary of the female. Why am I talking about seminiferous tubules and ovary? Because we are talking about gamete formation here. Okay, now if this cell were to undergo spermatogenesis, that means it undergoes meiosis, it is going to produce, you know that through meiosis you get a ratio of 1:4. 4 sperm are produced from one male germ cell, but it is going to produce two different types of sperm. So half of the sperm will carry X chromosome alone and half of them will carry Y chromosome alone. Why not both X and Y? Because the division is meiosis and the sperm are, now this cell is a diploid cell. That's why it has two copies of sex chromosomes. But the sperm are haploid, so they have only one copy, either X or Y. Okay, same thing for female here. This is a cell which is diploid sitting inside the ovary. Now this cell decides to undergo oogenesis, which is again involving reductional division or meiosis. All the eggs produced will carry X chromosome in them. Okay, so these are haploid again. Why only one X? Why not two X? Because these are haploid. They will have only one set of chromosomes. Similarly, they carry only one sex chromosome. In female, all the eggs will carry only one X chromosome. Clear? Now, if the sperm carrying the X chromosome fuses with the egg carrying the Y chromosome, the resulting zygote will be X with X. Zygote is obviously diploid. Similarly, if the sperm carrying Y chromosome fuses with an egg carrying X chromosome, the resulting zygote will be XY. Now this zygote with XX will become a female organism. The embryo becomes a female, and this one, which is XY, becomes a male, because having like sex chromosomes determines the female sex, having unlike sex chromosomes determines the male sex. Clear to all of you? Now tell me, what determined the sex of the baby, the sex of the zygote? Is it the egg or the sperm? It's the sperm, because there are two types of sperm. So what determines the sex of the zygote here? It is whether the sperm carrying X chromosome fuses with the egg, or whether the sperm carrying Y chromosome fuses with the egg. So the sex determining factor is not the egg, it is the sperm. So important thing to remember, in humans the sex is determined by the father. As you all know, if in people in rural areas and generally in olden days, we would hear about things and see things where people would blame the woman if she gave birth to a girl child, isn't it? Where the people have a lack of awareness, and but now you know, actually the one who is responsible for whether the embryo becomes a male or a female is not the mother, it's the father, because the male or the father is the one who is producing two types of sperm, whereas the female, the mother, is producing only, all eggs are of similar type because they all have only X chromosome in them. Okay, so this is just a simplified version of the explanation to give you the idea of sex determination in humans. Remember, I have focused only on the sex chromosomes and not on the autosomes. When you learn a chapter called genetics, you will probably learn about autosomes there. You will have a better understanding of that. Right now, in this chapter, suiting to the scope of this chapter, I have dealt with only the allosome part of it. Okay, fine. So now we have completed fertilization. In the ampulla, fertilization has happened, a zygote is formed. What is this zygote containing? The zygote has how many chromosomes? The zygote has, yes, 23 plus 23, 46 chromosomes, isn't it? So now we are going to see what happens to the zygote. The next step is called cleavage. The zygote is now sitting in the fallopian tube and it starts undergoing repeated mitotic divisions. By the time it starts moving through which part of the fallopian tube? Isthmus, the narrow portion. Remember, fertilization happened in the broader part, that is the ampulla. Yes, now the zygote has started making its journey through the isthmus of the oviduct. That is where the cleavage has started now. Okay, so I will draw a diagram to explain what exactly is happening here. Okay, so imagine this is the uterus and this is the fallopian tube and this is the fimbriae part of it. Okay, now somewhere here in the ampulla, and let me show the remaining part of the uterus as well, with the inner layer here, endometrium. Of course, the endometrium is present even in the fundus or the bulged part of the uterus, correct? Okay, now from here the secondary oocyte came in, and somewhere here, let me say ampulla, The secondary oocyte is sitting over here, and it got approached by many sperm, and fertilization took place, and now zygote is formed. Let me draw a bigger nucleus to the zygote here. Now immediately after the zygote is formed, so let us label this as zygote. Okay, now so after fertilization, zygote is formed. Now can you see how I'm showing here? As soon as the zygote is formed, it starts dividing. See, yes, are you able to see how the cells are increasing and as it divides? Yes, see, here the zygote has only one cell. It's a single cell. But now here it has become a two-celled embryo, and here it has become a four-celled embryo. Did you understand what I'm saying? And then it becomes an embryo with 8 cells as it further progresses, and by the time it has reached into the uterus, the embryo has reached into the uterus. Now this process where there is repeated mitotic divisions, the cells increase in their number. 1 becomes, so this is 1 becomes 2, and 2 becomes 4, and 4 becomes 8, and so on and so forth. This process is called cleavage. And what are these individual cells that are obtained by cleavage? These individual cells are called blastomeres. Please remember, the products of cleavage, the cells which are put out during cleavage, are referred to as blastomeres. So here there are 2 blastomeres, here there are 4 blastomeres, and then there are 8 blastomeres. And by the time the embryo has moved into the uterus, it becomes, see, it becomes a solid ball of cells with many cells in it. And this solid ball of cells, the embryo which has entered into the uterine cavity, is now referred to as a morula. The stage of this embryo is called a morula. And how many cells does it have? It has 8 to 16 blastomeres. And how did the number increase to such a great extent? The number increased to such a great extent because of cleavage, because of mitotic divisions. Yes or no? Now what happens is, this morula goes further down into the uterus, and it transforms into, it suddenly becomes hollow. Here it was a solid ball of cells. Now it becomes a hollow structure with cells on its, a layer of cells on its wall. See, it has a wall of cells, a single layer of cells in its wall, and it has a mass of cells inside. And this embryo is now called blastocyst. It has more than 16 blastomeres. Did you understand? So we started off with a secondary oocyte here. This is the secondary oocyte. And we saw the sperms come by and fertilize the secondary oocyte. And then a zygote is formed. And then we saw how the zygote undergoes repeated mitotic divisions called cleavage. When the zygote divides, first it becomes 2 cells. Then 2 cells divide and it becomes a set of 4 cells. 4 becomes 8, and this embryo, which now has 8 to 16 cells in it is called a morula. And what did I call the cells which are obtained as a byproduct of cleavage? They are called blastomeres. And this morula is a solid ball of cells. You don't see any cavity inside, do you? No. And then what happens to the morula? See here, the morula transforms into a hollow embryo. And what is this hollow embryo made up of? It's made up of a single layer of cells outside and a lump of cells inside, and there is a cavity. See, this cavity is called the blastocele. And this hollow embryo, so please bear in your mind that blastocyst is not a solid embryo, it is hollow. Which is the solid embryo? Morula is the solid embryo. Actually, morula is named after the scientific name of mulberry, Morus alba. It looks like a mulberry fruit. This solid cells which you see here, it looks like a mulberry fruit, and that's why it is called as morula. So let me cover the entire morula here. So this is the morula, and then the morula develops a cavity inside and it becomes what? Blastocyst. Cyst means a fluid filled structure. Since there is fluid inside, and since there is a layer of cells around it, and there is a lump of cells inside, we call this embryo as blastocyst. This blastocyst is something very, very important. I want all of you to be attentive and focus on the blastocyst, because it is this blastocyst which is finally going to attach to the endometrium of the uterus. See, this is the endo. I hope you all remember your basics. The inner glandular and vascular layer of the uterus, the inner wall of uterus is called endometrium. Clear till here? Okay. So we saw the zygote moves through the isthmus, it undergoes mitotic divisions, and when it undergoes mitotic division, what are the resulting cells called? Blastomeres. So we saw first we had a zygote, then the zygote became two-celled, then the two-celled became three-celled, four. So first it was one zygote, it became two cells, it became four cells, and then it became 8, and then it became 16. And this stage where it has 8 to 16 cells, like we saw, it is a solid embryo called morula. And what are these individual cells? These individual cells are referred to as blastomeres. The cells obtained by cleavage are called blastomeres. See, there's an increase in geometric progression, 2, 4, 8, 16. Now we had learnt the embryo which has now reached the uterine cavity, which has 8 to 16 blastomeres, is called morula. Division continues, cleavage continues, so that the morula will now have more than 16 blastomeres, somewhere between 32 to 64 blastomeres. And that cell stage is called a blastocyst. I had told you that the blastocyst is a hollow embryo. Okay, so this is the process of cleavage. Now, structure of blastocyst, I had all showed you a rough diagram of how the blastocyst looks like. Now we had seen that there is a layer of cells outside. We'll show nucleus for each of these cells. I'm going to label the parts for you here. This outermost layer is a single layered structure, and then I had told you that there is a lump of cells, a cluster of cells. Correct? So this outer layer of cells please take down, is called the trophoblast. The cavity is called the blastocele. It is actually filled with the fluid. And this lump of cells inside is called the inner cell mass. This is how a blastocyst looks like. There is an inner cell mass, there is a trophoblast. Yes or no? Now what is the uniqueness is, it is the trophoblast which actually attaches to what? To the endometrium, the inner lining of the uterus. And it is the inner cell mass which develops as embryo. The actual embryo, the human embryo, is derived from the inner cell mass, because this inner cell mass is made up of special cells called stem cells, which can give rise to all the cells in the embryo. Are you following what I'm saying? Yes? So blastocyst has two layers, or it has two, it is organized into two structures. An outermost layer of cells called trophoblast and an inner lump called inner cell mass. The inner cell mass has special cells called stem cells, and this inner cell mass gives rise to the actual embryo. The trophoblast is the one that attaches to the endometrium. And the trophoblast is what? Is it single layered or multi-layered? It is a single layer of cells. Outer layer. Is it clear? The structure of the blastocyst. Okay. See here, you can see the real blastocyst. See this outer layer here. The outer layer of cells is very clearly seen here. And see this lump of cells inside. This is the inner cell mass, isn't it? And this is the cavity, the blastocelic cavity. So it has trophoblast, it has inner cell mass. Trophoblast is the one that actually forms a connection called placenta, because this is the one that attaches to the endometrium of the uterus. You know that the developing young establishes an intimate connection with the mother's uterus, and this is called placenta. We will be learning in detail later, okay? And the inner cell mass differentiates and forms the embryo because it has stem cells in it. Alright? So this gives you, this is the real under the microscope view of a human blastocyst. Now coming to implantation. What happens the moment the blastocyst comes in contact with the uterine endometrium? See, this is the endometrium. This pink layer that you can see here is the endometrium, okay? And you can see how there are cells. Now this is the blastocyst. See here, this is the trophoblast, the outermost layer of cells. Correct? All of you agree with me? And this is the inner cell mass. Let me shade the inner cell mass for you. This is the lump of cells inside which actually gives rise to the embryo. The moment the blastocyst attaches to the uterus, it will undergo certain changes and it will differentiate into certain tissues, which I will be discussing a little while. But this attachment of the blastocyst to the uterine endometrium itself is called implantation. Now to give you a better explanation of this implantation, I have a blank slide here. So this is the uterine endometrium. Okay, now the blastocyst has attached, it has come in contact with the endometrium. You know that the blastocyst has a superficial area, a superficial layer here, the wall of the blastocyst. What is this layer called? Can you recall and tell me? It is called the trophoblast. Yes. and this is the lump of cells inside. Inner cell mass. Okay, now as soon as the blastocyst attaches itself to the endometrium of the uterus, now this is the uterine endometrium, you can see these folds in the inner wall. It has lot of folds and depressions, the endometrium of the uterus. Okay, as soon as it attaches, this trophoblast, this superficial layer I told you is the trophoblast, differentiates into two layers. Now I am showing you a layer here which has finger-like projections. So this layer, this tissue also comes out of the trophoblast. So this happens after the blastocyst has attached to the endometrium. Okay, so this tissue which has these finger-like projections is formed. When is it formed? Once the blastocyst attaches to the endometrium. So it's somewhat like this. If this is the blastocyst, it will develop a tissue which has finger-like projections, and this tissue will have multiple nuclei without boundaries between the nuclei. So it's a multinucleate mass of protoplasm or cytoplasm. Whereas the layer inside will still remain cellular. See, the trophoblast inside is cellular itself. Correct? And there is inner cell mass. See how it has developed this structure which have these finger-like structures, finger-like projections. And this structure which has multiple nuclei in it, this tissue is referred to as syncytio or syncytio trophoblast. Whereas the remaining cellular, what remains as a cellular structure, this inner layer of the trophoblast which still has cells in it, is called the cytotrophoblast. So this happens when the blastocyst attaches to, what is this inner layer? Endometrium of the uterus. So it's as if the trophoblast at the point of attachment, this is the site of attachment, at this point of attachment it differentiates into syncytiotrophoblast, and the remaining cellular part which remains as it is called the cytotrophoblast. And these finger-like projections, for your information, these finger-like projections are called the chorionic villi. I have labeled one, so villus. So the finger-like projections that are formed by the trophoblast on the uterus, on the blastocyst attaching to the endometrium, is called the chorionic villus. And then what happens is, this endometrium, the cells of the endometrium will start growing and they will overlap the blastocyst. See, they are covering the blastocyst. Why should the endometrium totally encapsulate the blastocyst like this? So that it gets firmly embedded. This process where the blastocyst gets firmly embedded in the endometrium and it gets engulfed, or it is covered by the cells of the endometrium, is called implantation. Do you understand? So this is nothing but cells of uterus, endometrium. It literally encapsulates blastocyst. See, it is encapsulating the blastocyst. The blastocyst is sitting safely inside now. It's buried in the uterine endometrium. All right. So I hope you understood the process of implantation with this. Let's move ahead to the next slide. What did we learn today? Now today we learned the meaning of fertilization, fusion of a sperm with an ovum to form a zygote. We learned the mechanism we learned arrival and movement of the sperm. Then we learned acrosomal reaction, entry of the sperm, karyogamy or amphimixis, where the nuclei come together and the chromosomal material merge. Then we learned how the sex determination happens in the case of humans. Then we learned the process of cleavage, yes, where a single cell in a zygote becomes an embryo with 2 cells, an embryo with 4 cells, an embryo with 8 cells, and so on. And then we call it names. We call this as morula, and further we call it as a blastocyst. We learned the structure of human blastocyst and how it attaches to the endometrium, which is called implantation. All right. Homework for today. The first question is a 3-mark question. Draw a neat labeled sketch of the ovum surrounded by a few sperm. Define the terms fertilization, cleavage, blastomeres, morula, implantation. I forgot to write marks here. 5 marks, 1 mark each. What induces the secondary oocyte to complete its meiotic division? Name the part of blastocyst that gets attached to the endometrium. Name the part of blastocyst which gets differentiated as the embryo. Name the 2 sex chromosomes which are found in the human male. So with this, we conclude the lecture for today. So all the very best, study well and do your homework and have a nice day. Until next time, take care all of you.